So Alex Nicols obtained his bachelor's in medical microbiology and a master's in molecular medicine from the University of Sheffield Medical School in the UK, where he studied the virulence factors of Burkholderia pseudomallei. He obtained a PhD in translational immunology from the Newcastle University Translational and Clinical Research Institute, where he worked closely with the Great North Children's Hospital, investigating interactions between the gut microbiome and peripheral immunity in reconstituting bone marrow transplant patients. He also worked as part of the PITCH consortium using high-dimensional cytometry to study T-cell memory responses to SARS-CoV-2 vaccination and infection. Alex is now a postdoctoral scholar in the Pediatric Immunization Advancement Lab at the University of California, San Diego. His research focuses on using high-dimensional spectral cytometry alongside metagenomics and metabolomics to better understand responses to vaccines in early life. So Alex, thank you for joining us today. And the title of your talk is Understanding the Impact of Hybrid Immunity on SARS-CoV-2 T-cell Memory. Over to you.
Thank you very much, and good morning, everybody. I'd like to say from sunny San Diego, it's actually a bit cloudy at the moment, but thank you for taking the time to listen to me, and thank you for the invite to speak today. So I'm actually going to speak today not about work that I'm doing in San Diego, but about work that I did in England as part of the PITCH consortium, and looking particularly at T cells and their response to COVID-19 vaccines in healthcare workers. So this is a small overview of the study. We recruited in the beginning around 2,000 healthcare workers across 5 different NHS sites up and down the country. That number's now reduced to about 600 that we're following regularly. And the data I'm presenting today particularly pertains to the first 3 doses of the Pfizer-BNT 16-2b2 vaccine and also Omicron breakthrough infection. So a little bit of background about what we'd found in PITCH with respect to T-cell immunity. So the first big paper we published was in Lancet, and that was led by Dr. Adriangial in Sheffield. And that was looking at the first dose and the impact of prior Wuhan infection on the magnitude of response after 1 dose. And rather unsurprisingly, but still important to show at the time, was that if you were infected prior to vaccination, you had higher T-cell responses. The second paper, which we published in Cell, which was led by my old supervisor, Dr. Becky Payne in Newcastle, this was looking at the second dose of vaccine. And the major question we had here at the time was what the difference in immunogenicity was between the standard and the extended dosing regimen. So that was 4 weeks versus 10-week gap between the first and second dose. And the main finding among lots, although antibody levels did wane after the first dose, memory B and T cell pools were well maintained. And then the third paper, which was led by Dr. Shirley Moore in Liverpool, published in Cell Med, and this looked at the third dose of vaccine. And here we found that although there was significant waning of the spike-specific T cells 6 months after the second dose, this waning was far, far less than that of peripheral antibody. And the T cell responses that were induced after a third dose of vaccine, importantly for what would happen subsequently, were cross-reactive to Omicron variants BA1 through BA5. And so all of that data was very interesting, but a lot of what we were doing in these papers was sort of using gold standard techniques, so ELISpot, ICS, basic ICS assays, and antibody assays. And one of the key things that we were interested in looking at further was the memory phenotypes that were induced by vaccination, and how they may evolve across vaccination, and particularly what the impact of prior infection was on shaping those phenotypes. And so I led a sub-study using MHC1, SARS-CoV-2, and EBV and CMV specific pentamers and dextramers, some of which came from ProImmune. And I combined this with spectral cytometry, high-dimensional cytometry being my sort of expertise, if you will. And the main goal of this was to really get a highly detailed profiling of what was going on in the quiescent memory phenotyping of these responding cells. And so What we're seeing in this slide is just raw magnitude data. So if you look at the top panel, this is just a representative flow cytometry plot of a particular participant across these 3 vaccine regimen. And this is how we can gate for MHC positive cells. And COVID-19 is quite nice to work with because it induces quite high responses relative to some other viruses. And so in this bottom plot here, you're seeing those magnitudes plotted. On the x-axis, we have time point relevant to the vaccines, and on the y-axis, we have the magnitude of SARS-CoV-2 spike-specific CD8 T cells as a proportion of the total CD8 T cell pool. So there's 3 things I really want to take from this first slide. The first and most obvious immediately is that, yes, indeed, if you had Wuhan infection baseline, your post-vaccine magnitudes are higher overall, particularly after 2 doses. But if we look a bit closer in this right-hand graph, which is looking more closely at the waning and boosting of the 3rd dose, we see that indeed, again, following what we found with ELISpot, the spike-specific T cells do indeed wane. And the 3rd dose does boost the T cells, but it only boosts them back to the levels that they were previously at by 2 doses. And this is true for both infection or non-infected vaccinees. And so this perhaps suggested that there may be some form of immunological set point with respect to the immunogenicity elicited by spike vaccine. And then if we looked a little bit closer, we noticed that actually in the previously Wuhan infected people, there was a small proportion of participants who had very high magnitude responses. So this is, we defined as having magnitude responses of greater than 1% of their total CD8 T cell pool. And you can sort of see them here at the top of the graph. And if we were actually to remove those participants, these red and black graphs would look pretty much identical. So the next thing we wanted to really look at, which was the crux of our research question, is what does this mean for phenotype? So based on what we'd found looking at the magnitude data, we split the cohort into these 3 groups. So the naives, and then the infected normals, which we defined as infected people who had peak magnitudes less than 1%, and then these infected high individuals. And what you're seeing here is just a median expression for all of the markers that we had in the spectral flow panel. But on the right, I've blown up the significant markers that were up or down, like higher or lower expression in these infected highs. So in this black square, we see that in these high magnitude responders, that CD62 ligand, CD28, and CD27 are all significantly less expressed as by a Kruskal-Wallis test. And then in the red square, we see that CD57 is significantly increased. And this sort of marker profile is indicative of a sort of drive towards terminal differentiation and senescence in these high magnitude responders. And so if we look at this a little bit closer, again, focusing on just a 1 month post-dose 3 time point. So what we can do is we can cluster these antigen-specific CD8 T cells based on that expression data that I showed in the previous slide. And what you're seeing here is a T-stochastic neighbor embedding visualization of those 15 clusters. And obviously, because we're looking at T cells and T cells only, obviously T cells are more similar to each other than they are different. So that's why these these kidney plots aren't as well separated and nice looking as they may be in a whole blood study where you have B cells and T cells and natural killer cells all very well defined. But there is still a pattern that emerges. And if we look at the infected high participants, in these red circles, we have some clusters that are seemingly highly, are in high abundance in these particular participants as compared to the infected normals and the naives. And if we look at actually what these clusters represent in terms of the markers that they have expressing on them, you can see that they're different variations of T effector memory RA cells and effector memory 3 and effector memory 2 cells. But the thing that they all share is that they're all CD28 negative and CD57 positive. And then in these black circles, we have clusters that are seemingly pretty much absent from these infected high participants. And again, these represent T stem cell memory cells at different stages of early or late activation in memory. And so if we graph this up, what we can do is we can export the frequencies of these clusters and just plot them in a graph that's just easier to visualize. So what you're seeing here on the y-axis is the frequency of each cluster as a proportion of the number of antigen-specific CD8 T cells to spikes that each patient had. And so what I've done is I've grouped them slightly just for ease, but you can see hopefully that these CD28 negative, CD57 positive effector memory cells are significantly higher in abundance compared in the infected highs compared to the other two groups, with the CD28 negative, CD57 positive telomeres being the most highly induced. So this data was very nice, and we were getting quite excited about it, and we were planning to sort of expand it to the full cohort. But around the same time that we were doing this was when the Omicron variants were becoming sort of the dominant strains, and we were getting a lot of breakthrough infections, and everyone was getting kind of worried. And then a couple of papers were published that showed that actually Omicron infection and the immunogenicity that's elicited by Omicron infection may actually be muted in people with previous infection. And so this sort of caught our attention, and obviously having this quite nice platform that we developed, we wanted to then apply that to that research question. And so again, what you're seeing here is magnitude, again, just as a proportion of CD8 T cells. And again, the blacks representing the infection naive, and so with respect to Wuhan, and the red showing people who were previously Wuhan infected, with the open and closed circles being the pre-Omicron, post-Omicron time points, respectively. And what we actually noticed on this left-hand panel, which is the magnitude of spike-specific CD8 T cells, is that, yes, indeed, there does seem to be some form of imprinting with respect to previous Wuhan infection, in that Omicron infection doesn't seem to elicit a magnitude increase in the previously infected, whilst it does in the infection naive. But we then actually expanded this question out, and we looked at non-spike specific T cells using multimers directed against nucleocapsid proteins in particular. And here we see that that's not the story for non-spike, and actually Omicron infection does elicit a magnitude increase, irrespective of prior infection status, in terms of the non-spike specific T cells. So, we hypothesize here that this is actually, going to be beneficial for protection going forward. And all of this data is published in our paper we put in Nature Communications. And the T cell work we did in that paper was obviously part of a greater study which looked at antibodies and neutralization and B cell memory. And so with that in mind, we wanted to sort of streamline the T cell phenotyping. And so we looked particularly at 6 sort of canonical memory phenotypes that are pretty well defined, those being naives, stem cell memories, central memories, transitional memories, effector memories, and then these terminal effector cells, which are the same as those CD28 negative, CD57 positive telomeres that you saw previously. And again, we saw a similar pattern here, that the previously Wuhan infected had a large dominance of these terminal effector cells compared to the previously Wuhan negative. And again, we actually noticed that Omicron infection, not only does it not elicit a magnitude change with respect to spike, it doesn't really elicit a phenotypic change either. So this really reinforced for us that there is some form of imprinting and set point with respect to spike-specific CD8 T cells in people who were previously Wuhan infected. But if we look at the non-spike immunity, there is actually, again, evidence of phenotypic evolution, but it does seem to be following a similar pattern to spike. So what's particularly interesting in my mind, and this is obviously something we can look at with this cohort we keep going, is if there is multiple Omicron or multiple infections, is this non-spike immunity going to follow the same pattern that spike does and eventually reach a set point for people who were previously Wuhan infected. So that's going to be important to look at in the future. And then one thing we sort of added on out of just interest and discussion was we included a sort of semi-functional output to these multimere-bound cells by including intracellular granzyme B staining. And it turned out to be one of the most striking findings we found, and we saw that actually in the people who were previously Wuhan infected at baseline, they had vastly higher granzyme B content in their spike-specific CD8 T cells as compared to the uninfected. But again, the Omicron infection didn't seem to change that, really. So that was quite a quick tour of quite a lot of about 3 years worth of work, but hopefully I've shown that vaccination after prior ancestral infection results in significantly higher spike-specific CD8 T cell magnitudes, that T cells do indeed wane 6 months after the second dose. They are boosted back by the third dose, but this boosting is limited to levels previously established by a second dose. That effector memory and effector memory RA cells dominate the CD8 T cell memory pool to spike, with previous Wuhan infection actually resulting in a significantly higher cytotoxic potential. And high responders, which is driven exclusively by prior Wuhan infection, display a definite shift towards terminal differentiation and senescence-like states. And importantly, going forward for the way the world's going, spike-specific immunity by Omicron, although it is muted, non-spike immunity does continue to be boosted. But as I said, it's important to monitor that non-spike immunity to make sure it doesn't reach a plateau in the same way that spike does. And with this very lengthy acknowledgement slide, I thank you for your time and open the floor to any questions you may have.
Great, thank you very much indeed, Alex. Great to hear that. It's a lot of exciting data there from a time which we all, in a sense, want to sort of bury in our minds, but at the same time, there's a lot to learn from it. So on that point, while people are thinking of any questions that they'd like to ask you, I'd like to maybe kick off. And just in the context of understanding the kinetics of what we've seen here with re-exposure to follow-on strains of SARS-CoV-2, how can we apply this knowledge to? Is this knowledge only really applicable to this virus, or is it something that other more broad learnings that we can think of in the context of what we've been able to investigate during the pandemic? Is there something that we can apply this to other viral infections that might come along?
Yeah, I think definitely. I mean, one of the things that we're particularly interested in, even with respect to COVID now, is obviously in pediatrics, we're seeing what happens to babies that are born who have never seen Wuhan, and now they're going to be infected by a totally different strain, whether this pattern is the same. But with respect to kind of future proofing for future pandemics, these sort of analyses that we've developed very quickly over time, we now have these tools available to us. And so with this particular panel, this can be applied to essentially any virus you want. You just have to swap out the tetramers or the multimers or the pentamers to look at whatever you decide. But particularly with respect to antigenic sin, and that's been around for since 1920 or whenever it was first described. So it's not necessarily new in terms of medicine. But one of the things I think that we've gleaned from the COVID pandemic and all of these studies into longitudinal T cell phenotypes and what that may mean in terms of long COVID and protection going forward, is that in future responses to emerging viruses, that the vaccine design strategy needs to focus on more than one facet of that particular infection. So the problem, I think what we're seeing here is an artifact of the fact that people were infected with a Wuhan spike, and then they got subsequent vaccinations with the exact same immunogen, whereas they may benefit from having spike vaccination, non-spike vaccination, or at least multiple different versions. And the other thing, of course, which is becoming increasingly popular is, what's going on in the mucosa and the separation between mucosal immunity and peripheral immunity that we're looking at here, and how we can better induce mucosal immunity to protect against these viruses.
Great. Yeah, thank you very much for explaining that. Perfect. Are there any questions in the final minute or so? It seems not. Okay, well, thank you again, Alex, for your time and joining us from not so sunny San Diego, but hopefully the sun picks up later.